Okay, so good morning, everyone. Let's start. So I'm Juan Restrepo. I'm a PhD student at Cox Lab as well, and we'll be talking about Perseus for 3 hours. So it will be a lot of fun, and we will start with a short introduction to the theory behind it. So like all the statistical models that we use and all the math concepts behind it, and then we will exactly. Do like a hands-on presentation, let's say, or training about how to get like how to use the basic features of it, and then more advanced one. So, as Jürgen and my other colleagues mentioned in the first day, so we actually have two softwares. One of them is MaxQuant that basically takes raw files and convert them to protein expression profiles or basically a matrix with numbers that represent the intensity and the proteins that exist in your sample. And the other one is Perseus, that is to analyze these output tables, let's say. So what Perseus does is that it offers a set of tools that can be exploited by bioinformaticians in an easy way to convert your data or like, let's say your matrix with numbers into information. So this was the original publication here on the left. It was developed at Cox Lab, led by, of course, Jürgen and Steph Katianova. She's actually now in the Bay Area in California, working in bioinformatics, but she started in our group. And then exactly, it's a software that is very widely used and exactly. So in principle, you could even like analyze every type of data with it, but normally it's used to analyze omics data. So what does Perseus have? What can you find there? You can find a set of like different sets of tools to do statistical analysis. So this means like PCA and T, like to sample t-tests and more. So like everything you need to like, I don't know, do expression analysis and like differential expression and all of this. You can analyze multi-omics data with it. So at the, let's say, transcriptomics and proteomics at the same time, or whatever you can think of. You can use like a very wide range of visualization tools that include scatter plots and heat maps for clustering. There is a sort of specialized module that includes machine learning tools and also exactly enrichment analysis, even at the multi-omics level. And of course, you can do a lot of exploratory analysis, which means like data filtering, imputation, yeah, histogram visualization, all of this. And everything is built on top of a matrix. So like this is the data type we use. We use a matrix and then we extend it with annotations, let's say, from Go or Keck or whatever like it's better for you. And it also has like a set of special columns that can be text, let's say the gene names or the protein identifiers. But then everything is like analyzing a matrix, which can be in expression proteomics, the protein groups table that MaxQuant gives you. So besides the original publication, these are two very nice, very interesting and clear manuscripts that allow you to get deeper into MaxQuant, Perseus, and also shotgun proteomics in general. So the one on the left is actually the first group, the first paper I read about Cox Lab when I joined. So it's a very nice review that like really walks you through shotgun proteomics. And goes all the way, like it starts from the data preparation and it goes all the way to data analysis with Perseus. So data preparation, then the experimental setup of a typical proteomics experiment, then MaxQuant, and then Perseus. And on the right, this is sort of more like more tutorial-like paper that will really take you step by step through like what you can do with Perseus. Okay, so exactly. So as I said at the beginning, Perseus in principle can be used to analyze any type of data. So you could analyze real estate prices, let's say in Boston, for example, but it's normally used for omics data. So one very common use case of Perseus is expression proteomics. So what can, and we have a specialized set of tools for dealing with this type of data. So what can you do with it in Perseus? So first, you have to load in your data, right? And this will typically, in expression proteomics, this will be the proteins group table. And this will have, let's say, I don't know, the LFQ intensities, or even it can also be, let's say, if you are talking about, if you are using it to analyze genomics data, it could be like a counts matrix or whatever. But the important thing is that you have numbers that represent the intensity in the entries of the matrix, and then you have samples in the columns, and then you have proteins in the rows, right? Exactly. So first, you upload the data, and then you want to do probably some pre-processing. This means like filtering out contaminants, decoy matches, and things like this. And then you probably want to also filter out the rows that had too many zeros. So they were not measured in many samples. And then you will probably want to do imputation. So you can do this natively just by clicking in a few buttons, and we will see how to do it later. You can also do some data exploration, let's say with PCA. This is typically done this is typically done also at the beginning of your project to really see that the samples actually separate correctly and there is nothing wrong, there is like not batch effect or something like this. You can also do a differential expression test, and you can see it here. You can visualize it with a volcano plot. You can do it for 2 samples, and like the volcano plot is of course used in this case, but you can also do it for like comparing more than 2 samples, and this is called the ANOVA, and we will do it here later. Exactly. You also can use Perseus for clustering. So we implemented a hierarchical clustering algorithm in Perseus. It's also very easy to use. You just have to click on the icon, and it will give you a nice heat map that you can read in a very, let's say, personalized fashion, right? So like you can also tune it and personalize it. So you can also see here that it actually clusters at 2 levels, at the samples level and at the proteins level, which is quite useful. You, of course, can do the annotation enrichment. So in order to see how, like which biological pathways were more active or less active in your experiment. And you can also do it at the multi-omics level. Of course, we also provide time series support for circadian rhythm studies or exactly things like this. Exactly. Another use case will be the post-translational modifications study. So like now, we are not talking about spatial expression proteomics, but some kind of, let's say, phosphorylation. And in this use case, the matrix is also very similar to the one before, in the sense that you still have a matrix with numbers, but then the meaning of each row is different. So each row is not a protein anymore, but a phosphorylation, a peptide, but a phosphorylation site. Yes, exactly. So in this particular use case, you might be interested in enriching your data with annotations. And you have a specialized database as PhosphoPlus, and you would like to basically pull the annotations there probably. So exactly. So we can also help with that. So it's exactly, this is like a very common use case, and we implemented a plugin to do this easily. You can also put, it doesn't have to be phosphosite plot, right? It can also be a UniProt or another database. You can also use Venn diagram visualization of your data. Exactly, just by clicking a few buttons. And this is particularly important to either compare two samples or see how does a specific subset of your phosphorylation sites, let's say your peptides, actually how big they are compared to the total population. You can also do motif analysis, meaning what are the amino acids that, let's say you define a center of your window, and then you also define a radius, and then you want to see which specific amino acids in every position will be more useful for like really activating this site that is in the center of the window. Exactly, and then you can also do like normal visualization and imputation for this type of data. Yes, so we also, as I said at the beginning, we also provide a learning module. So exactly in machine learning, what you want to do is basically learn from your from the data you have and create a rule that will generalize better for like unseen examples. So like in this example, let's say the blue and the, I think, pink or purple points will be two different classes, right? So this will be, let's say, healthy, and this will be sick. And then you have like a ground truth data. So this means like you have examples, like you basically have expression profile across samples of the proteins, and then you say, okay, this one is healthy, this one is not healthy, and then you train let's say a classifier for this. And then the idea is that later you can use this classifier to, just by measuring the expression profile of an unseen and undiagnosed patient, just to see if he's actually healthy or sick. Exactly, and this particular algorithm is called support vector machine. It's normally, when it's like in its most simple version, is actually flat. So the separation plane is flat, but you can also add a kernel to make it curvy. Exactly, there are many parameters for this. Okay, exactly. So this will be one use case of machine learning genomics analysis. So let's say predicting exactly the state of a system given the proteomics expression. Another use case will actually be different. It's more related to the differential expression test. So this is basically what you can see here in the middle is an alternative way of finding a set of differentially expressed proteins or peptides that are part of a protein, of course. So the idea is that you basically do the same as you did before. You train to predict healthy or sick, and then at the end you analyze your model, your classifier. So like you basically like look into this black box and see which subset of proteins were more important for reaching that prediction, and then you choose 5, let's say, or 6 or 7, and then you claim, these proteins are actually markers, right? So these are actually the proteins that actually look at to really have a good diagnosis. So this will be the second use case. The third one will be the analysis. So like you can do let's say some kind of classification, but they're not at the sample level, but at the protein level. So now you don't have like 5 or 6 samples, but you have 10,000 proteins or 20,000 proteins. And then you want to see like their variation across samples. And then you actually want to infer properties of the protein. And this can be in particular subcellular localization, let's say. Exactly. So the idea is that actually if the proteins have something in common, they should actually like cluster together somehow. They should be together somehow. So we also offer a set of tools for biological networks. There will be a complete presentation about this, so I will actually not go into a lot of details here. But yeah, the idea is that we offer basically two methods for building up networks. One of them is for building it from scratch, so like the whole interactome experiment, and the other one is just by like pooling an already constructed network that is very well curated. It's very famous, it's very complete. So you can also pull it and analyze it in Perseus. And then you can also, and this is a development that it was done by Hamid, if I'm not mistaken, a colleague of us in our group. So you can also like search this large network in the context of your experiment. So you pull the network and then you analyze it in the context of your experiment. But we'll see about it in another presentation, I think, with Carlo. Exactly. So as I said before, you also can do cross-omics data analysis. So it's not only proteomics, you can actually do it in any types of omics. Exactly. So let's say here you have actually 3 different types of omics. You have translatomics, transcriptomics, and proteomics. And here you are basically simply visualizing how correlated they are. So you match, let's say, an mRNA strand to a specific protein, and then you plot in this 2-dimensional plane exactly like the coordinates of the intensity, and then you see how correlated they are. So let's say if it was like perfect correlation between, let's say, transcriptomics or proteomics, they will be like completely like a straight line, and the R square will be 1. But then exactly, so this actually means that there is a good reason to perform different omics because the correlation is not 1. Exactly. And then here on the right, you can see expression, so enrichment analysis at the multi-omics level. So you basically do the enrichment analysis at the protein level. You get a score. You also do it at the transcriptomics level. You get another score, and then you match a protein to a RNA strand, and then you plot it in this 2D plane. And the important thing, or the most interesting thing, is that, like, okay, most of the pathways are actually enriched or depleted in both, but some of them can actually only be seen in one omics. So it's already like a good argument to use it, or like you will only see it there. Okay. And then we also support cross-omics data analysis with time series, for example, for circadian rhythm. Okay. So if you have any questions, just feel free to ask, right? So no problem. So exactly, this is how the Perseus user interface looks like. So so this data is not important, right? It's just a random data from a Gaussian distribution. So it's not very interesting, but the important part here is like to see like how it looks like. So you get, of course, this very nice user interface, which is subdivided in like tools in the upper part, and then workflow in the bottom part. So the tools are also subdivided in 3 main groups. One is a tool, let's say a set of tools for loading. The second one, the largest one, is for pre-processing. So let's say the first tutorial, in the first part of the hands-on tutorial, we will be mostly using this one. And then you also have another one for analysis, which is like all the set of tools for plotting and like clustering, PCA, all of these. Exactly. So this will be like the set of tools, right? And then the bottom will be like the workflow. And the workflow is actually dynamic, so it changes. So it can have normally, so by default, it has actually 3 windows. The window in the middle has all the, so like shows you exactly the workflow of the data. So let's say the data pipeline. So like the process you have done. So it will start normally from loading the data. So like exactly. So you have a box with actions, and then you have an output, and then you have another action, and then you have another output, an action and output. So it will be like a chain. And then it like the complete workflow is like represented here, and you can go back to a previous step and then just click on it and visualize it. And then on the right is basically a summary of what you did. So if you click on this yellow box, it will appear here that you click on generate random matrix and the parameters you used. So it's a very nice way of like, okay, yeah, this is what I did, and I want to try something else. Then I just change it a little bit and run it again. And then on the left, you have either the out, so like, and on the left, you have the output of the action. So either the data or some kind of plot. Okay. So as I said, we have a workflow representation of the processing pipeline you have applied. So it will be this window right here, and then this is how it looks like schematically. So exactly, you start from an initial data matrix, then you apply an action, and then you get another, you get an output. And then this output, exactly, you can either continue applying actions, or let's say, oh yes, I apply, I don't know, I did a, what do I know, a filtering. I filter on valid values with the 70% threshold here. But then I was, okay, let's see how it looks like with 80%. Then I just branch out. So I select this matrix, I click on the action I want to apply, and I just change the parameter values, and it will give me another matrix. Exactly. And then you can also sometimes have actually one action that gives you two different matrices or two different outputs. Let's say a NOVAT, I think it gives you, okay, it might give you two different matrices, one of them with the transformed data and the other one with a p-value or another important information. And this is why it will give you two different outputs. Exactly. Yeah, and then you have a lot of combinations of things in between. Okay, so as I said in the first slide, what is really going through all of this is a matrix. So at the end, you are really like just applying functions to a matrix. And this is like what the main matrix looks like. So you have a set of intensities here, and this is the main matrix, so the one you are really applying your transformations to. And then you have enrichment, right? Or you have another set of columns. So you have like metadata. So you'll probably have some kind of column, right? If you control like that, or not probably, like you will have a column telling you if it was like part of if the sample is a control sample or in the stimulus side, right? Like you will have it here annotated like below the main matrix. Exactly. Or if you had replicates, then it will say, I don't know, replicate 1, replicate 2, replicate 3. But you will see it later as well. And then you also have exactly annotations. They can be text-based normally. And then you also will have numerical columns that could be like properties of the matrix, as like exactly the sequence length or whatever. And then you will have also like not categorical columns as these ones, but text columns. Okay, so something very nice about Perseus is that we, let's say, developed it in a way that the user can extend the software. So when you want to implement something, I mean, like, let's say you want a very specific algorithm that you are using or you are developing, and you want to use it in Perseus, then you can actually do it because we provide plugin support. So you can develop your code, do a few steps that we clarify, and then just use it inside of Perseus. And then we have a tutorial of how to do it here. So if you are interested, just go here. So how do you do it? Like you look exactly. So like how can you so like what do you need to know, let's say, to create these plugins? So you have to you have to, of course, know how to program, but you can actually do it in C#, in Python, or in R. And what this means is that you can actually bring all of these algorithms, like the whole R universe, to Perseus, like with a defined set of steps. So exactly. So I will advertise this again. So if you are interested, go here, and this has already all the information. Okay. So this was more or less the introduction about like what the software can do, what are the main references, what are the specific use cases. Now we will start with the theory, like the statistical theory behind it. So this will be like the more, let's say, the more precise, like math driven, or like the more precise part, right? Like where you can like really understand what is behind it. So feel free to ask. I will try to go slowly here. Feel free to ask, and exactly, just don't just believe that everything is like that. If you have any question, this is the moment. Okay, so in proteomics.
Hey,
Yes.
sorry.
Yes.
I had two questions. The first one about the annotation enrichment that you mentioned. How easy it is to implement a new pathway or like a group of genes that I want to integrate into Perseus? Or does it only use like known pathways? They're already annotated.
Yeah, so normally we actually pull the annotations from, I will show you later. So we actually provide the annotations. So I don't know, Jürgen, maybe you know how, if one really wants to like add new annotations, how should one do it? Like,
yeah, I think we probably see better later when you show it
yeah.
live. And you can also use your own annotations. I mean, they are read in from text files into Perseus. And as Juan says, we provide these text files, but you could also edit or change these text files that are storing the annotation. So you could use other annotation as well.
Okay, thank you. Another question I had about the crossomics analysis is how does Perseus deal with missing IDs between protein and RNA? Like, let's say you have a protein to identify, UniProt has an ID, but if I try to use ensemble, there's no ensemble ID for that same gene.
Then there would be no mapping happening. So we usually do the mapping with the proteomics identifiers as the base, and then to each proteomic identifier of a protein group, and then we take the majority protein ID. We map then whatever fits from the transcript IDs, basically. And if there's no fitting transcript ID, then it would be empty.
I see. So we kind of lose that information.
Yeah, in that case, yes. But usually that's not the problem. Usually you always find a transcript when you found the protein. It's more the other way around that you lose information.
Okay, thank you.
Okay, so exactly. Let's say you have a expression proteomics experiment, and you want to identify, or you want to get like a set of differentially expressed proteins in your experiment. So for this, you will have a control and a stimulus. So let's say healthy sick patient, and then you want to see like exactly which proteins are like the biomarkers here, and then you like what like the thinking behind this is really like, or the question that you really want to ask is like, like which protein is really changing after applying some stimulus, okay? And then the question is like, okay, but all of them are changing probably a little bit, right? This changes a little bit from here to here, like between replicates, they change a little bit. And then after the stimulus, they probably change more, but then they also change between replicates after applying the stimulus. So like, when are they really different, right? Like when is this change meaningful? This is the question that like is represented in the, that is answered in the differential expression test. When are the changes between replicates compared to between groups meaningful? Okay, so exactly. Let's say you have a few replicates for the control and a few replicates for the stimulus, and then you are actually looking at 3 different proteins. So one of them changes a little bit within the control and the stimulus, respectively, but it changes a lot across them. Okay, so within groups it changes a little bit, across group changes a lot. Then you have a case in between, right? Like it changes, it changes appreciably within groups, and it also changes a lot across groups. And then you have one that changes a lot within groups and also a lot across groups. So the question is like, are they, so like which one of them is more differentially expressed? So like by visual inspection, you probably all will say, okay, the protein B is actually more differentially expressed. So like the change is more meaningful than the one of protein C, just by visual inspection. But then, of course, you cannot look at all the profiles for like your 10,000 proteins and do this. And then at some point it will get confusing, like in the protein A case, you might say, okay, I mean, like half of the people will say yes and half of the people will say no, right? So like, how do you sort this out? So you need to like make it more concrete math-wise, right? So in a way that you don't have to like take the subjectivity out of it. So it's also interesting. So this example is also interesting because all of them have the same mean within groups and the same mean across groups, right? Sorry, so the same meaning in both groups, and of course the same change. So the question is like, how can I make this more mathematically concrete? And the answer is the t-test. Exactly. So the t-test basically quantifies exactly what we said visually. So it's basically a measure of weighted variance. So first, you calculate the variance between groups. So this will be the red line right here. So like simply take mean in group 1 and mean in group 2 and subtract them, and this will give you the numerator. And then you also calculate the variance within groups. So now not the mean, the difference of means, but really the standard deviation, like not the square of standard deviation, right? So like the variance. Yeah, exactly. And then you exactly take the ratio, and then this is this is already telling you how much is changing a protein for a specific or exactly a profile, let's say, in a specific set of conditions. Exactly. So you do this for many. Exactly. So you do this for many proteins, for 10,000 proteins or whatever your sample has, and then you still want to know, okay, which ones are. So like I calculate this for all of them, and then you want to see like, okay, then how can I go one step further, right? Like what does it mean? So you also have to, I mean, there is a whole mathematical model around it, and then you do some distribution, do some fancy things, and then this is the part where it's a little bit subjective, and then you have to define what is significant for you. And normally significant is given by a p-value that is small enough. And like as p-value is small enough, is normally 0.05 or 0.01, depending on like how strict you want to be. And one could say, okay, this is really the end of it. But it's actually not, it's just half of the story, because the p-value is a probability. And a probability means that it's not like for sure that it will happen, but like sometimes, so it is, so the way you interpret like a probability of 1% is, if I run the experiment 1 billion times, then 1% of this will have this characteristic, but it's not like, so it has to be in long numbers, right? Like in big numbers. So like if you do the experiment many times, then you are expecting this ratio to take place. Exactly. And then in this case, we actually have the following problem that is well known in proteomics and in data analysis in general, and is a multiplicity problem. And is that because this is a probability, you also you will have p-values that are 0 points that are like under your significance value, so that you will think that these proteins are differentially expressed just by chance, right? And then you have doing the experiment 10,000 times, as is typical, then you probably have problems associated with it. Exactly. So this problem is known as the multiplicity problem, and then is very important because it increases chances of false positives. So you have to control for this effect. Exactly. So if you have 10,000 proteins and you have a significance value of 0.01, so you have to conduct 10,000 t-tests, and this means that by chance you will have actually 100 proteins that are significant just randomly. So like you should actually get a value of 0 right here, but you are getting 100 proteins that are differentially expressed due to the multiple hypothesis problem. So this is an illustration of like that this is the effect. And then we have to correct for this effect. And then the way we correct for this effect, so I mean, there are a few alternatives. Yes, exactly. So individual p-values are not significant findings, and then we have to correct for this effect. And then there are a few different methods to do that. It's the family-wise type of methods and the false discovery rate types of methods. And this is the one we use. And I will be explaining it now. Exactly. So the first one, the family-wise error rate, is basically, yeah, it's very conservative, very strict, and then you will lose a lot of information. And it's basically taking your significance value and dividing it by the number of tests you made, right? And this is like, okay, if I made 10,000 tests and I have a significant, and I define it a significance value of 0.1, then I will take this 0.01 actually, and then divide it by 10,000, and this will be my new significant value. And then you can do it, of course, but then you will lose a lot of information. So fortunately, there is another way to do it, and it's just like you are not controlling the number of false positives, but you are actually controlling the proportion of false discoveries. And this is the one we use. Is more powerful. You don't lose that much information, and you still get very meaningful results. Exactly. So an example of the first one will be the Bonferroni correction. An example of the second one, or two examples of the second one, will be the Benjamin-Hochberg correction or the permutation-based test. This is actually not a correction, but like a completely different test. So exactly. So let's talk about this. So false discovery, Benjamin-Hochberg. So it's similar to the Bonferroni correction, but in a local way. So you still take your significant value, let's say 0.01. You divide it by the number of tests you make, in this case 10, because you have 10 rows, so 10 proteins, let's say. So you take your significant value, you define by the number of tests, but then you also multiply it by the position you are in the table. And this is already like giving you a better approximation. It's sort of a local Bonferroni correction. Okay.
We have a question here.
Yes.
Does Perseus have statistical analysis to do stats tests on non-normally distributed data? Because the assumption of a t-test is that your data is normally distributed, but obviously, you know, you can test for normal distribution and your data may not necessarily be normally distributed. So does Perseus have tools for non-normally distributed data?
Yeah, so thank you so much for that question. The answer is yes, and it's the second slide, the next slide. So it's actually the permutation-based FDR test, and it's the one we use by default.
Thank you.
Yeah. Thank you for the question, actually. Exactly. Yeah. So as the colleague said, so the problem here is that this assumes normality, right? And so you can still do it sometimes, right? But you have to be sure that your distribution is actually following has a Gaussian shape. But we also have another alternative test, which is the permutation-based FDR test, and this does not assume normality. So you can do this like you can just do it like without really thinking a lot, because it's very robust. Exactly. So like, how does it work? So you have your control and stimulus sets, and then you also have exactly so replicates and then different proteins here. And then what you what you are going to do is basically like interchange one column from the control, one or two or three, but let's say one column from the control and one column from the stimulus, and then run your test again. Okay, so you take your first, you start with the data as it is. So you take like your normal data, you calculate the test statistics and the p-value, record this, like write it down in a sheet of paper next to you, then permutate two columns, then do it again. So calculate the t-test and the p-value, write it down below the one before, and then you do this as many times, I mean, in principle, as many times as you can, but normally it's actually just until you have a significant sample size. And then you record all of these values, and then you plot a distribution, right? And then with this distribution, because you know which points were actually false, because you know the ground truth, then you estimate your false discovery rate like this. Yeah, so it's like a direct estimation of the false discovery rate. You actually like do it with the data you have, and then you just know the ground truth, and then you just assume that this is the case in general, which of course makes sense. Okay, thank you. Okay.
We have a question, I think.
Yes.
So I'm wondering, for biological replicates, if you have like time series data and gaps in some of that data, are there ways to account for that?
Yes. Yes. So they for sure are. We will see them later. Imputation will be one, which also has some assumptions, but I will normally like filter out the data that has too many missing values and then do imputation. This is what I will do. I don't know if you have a comment. It's okay. Yeah, but actually we will do it. So in the first tutorial we will do it, and if you have more questions, then just feel free to ask.
Thank you.
Yeah.
Any other questions?
So I'll continue. Exactly. So let's assume that we run our test, and now we have a bunch of p-values and like absolute differences and t-statistics, right? So like we have the whole information. So we want to visualize it. Let's say the simplest case, you are comparing just two samples. So you want to visualize it. And then the best way of doing it, or like a very practical, I don't know if the best, but like the standard way, very practical one, is the volcano plot. So what can you see in the volcano plot? So because you have two samples, you can, of course, very easily calculate the difference. You just subtract the values you have. And then this is this is the x-axis. So like the difference across samples. And then on the y-axis, you don't have the p-value, you have the minus log of the p-value. Okay, so as we said before, and probably some of you know, or most of you, I don't know, the p-value, when the smaller the better. Okay, and then when you when you take the log of something small, so like close to 0, let's say, then this is a very negative number. So like, let's say the log 10 of 0.01 is actually I don't know, 100, I think. It's very small. Exactly. Oh yes, 2. Sorry, it's -2. Sorry. Yeah, the log of 0.01 is -2, and then the log of 0.001 is -3, and exactly. So this is and then you multiply it by minus, so like then it will be like actually something very positive. Exactly, very positive and very large. So like large values in this axis are good, are something good. So then the idea is like big differences and then big values are the best. So like this here is like very differentially expressed. It's very different, let's say. And things close to 0 are not significant. I mean, it's like basically nothing changes. And then, so if you exactly define an alpha, you can calculate the minus log of that alpha. In this case, 0.05. And then this defines a region, like a border between your differentially expressed region and your non-differentially expressed region. So like everything above this value right here is differentially expressed, and everything below is not differentially expressed. Exactly. Exactly. So as we said at the beginning, so this is a different. So like what the t-test really measures is like the change across samples weighted by the change within samples, right? So it's the mean, so the difference of the means divided by the variance. Okay. So exactly, I think I was a little bit mistaken. Sorry, I realized it later. I say the square, but it's not a square. It's actually a square root, right? So I mean, as normally, like the variance is the square root of the sum of the squares of the distances, and then you also have to account for the background variability. So one thing would be to exactly you calculate the standard deviation within groups, but then you also have another source of uncertainty, which is like basically exactly like what we call here the background variability, which is essentially I mean, you have to take into account that like experiments are not perfect, right? So and like there are pressure gradients, there are temperature gradients. Someone might have entered the room and the machine moved a bit, right? So you have to take everything into account. And then the way you do that is by adding the background variability. Okay, so it's basically just a constant term that we have optimized for you. So you don't, I mean, so like you can actually change it, but like don't do it if you don't have a good reason. But then this is actually, it makes a lot of sense to have it. So the effect on this of this term is to change the boundary of the region from being a straight line to being hyperbolic. Okay. So normally I think the default value is 0.1. We will see it later, but I think this is the value. Exactly. And then like, let's say if it was like very large, this will be close to a vertical line. If it's 0, as you can see, is like a flat separator. And then this is really, so like, but then the interpretation of the plot is exactly the same. So everything like above the separation line is differentially expressed, and then in between, or well, here it would be below, and then here would be like in between, is not differentially expressed. Exactly. So exactly. So like after having the set of differentially expressed proteins, how did we, so like what is that telling us? Like which biological pathways are enriched or depleted in our sample, which is basically combining two things. It's combining annotations and combining differential expression results, right? So like if we have a set of differentially expressed proteins that have a set like a set of given annotations, what can we say about the biological pathways? What is depleted or enriched? And this is, I mean, as you may know, is called the Fisher. Normally, is done. It's called like what is normally done for this is like to carry out the Fisher exact test, and then this is also this is something that is also implemented in Perseus, and it's, I mean, when you think about it, it's actually a not so easy to answer, because it's like, okay, yes, we have something significant, but do we have enough to claim that something is that a biological pathway is enriched or depleted? So it's really like a measure of how associated is your subset of or your set of differentially expressed proteins to a set of annotations. So when can you say this exactly? And then it's a lot of computations that you have to carry out. You have to do many contingency tables, and you have to do many computations, and then at the end you have just a set of biological pathways, a score, and then a plus sign or like a check mark when something is exactly enriched or depleted, and we will also see how to do it later. But I mean, it's like we have that already implemented, right? So you don't have to do anything that other just than clicking on a few icons. Exactly. So this is how it will look like the results of the Fisher exact test. So you will start probably with exactly. So here on the right, you will on the left, sorry, you will have if it's a Go term or a cake, or and like what is the category value of it, and then also the enrichment factor, right? If it's like big, then it's like exactly like this one is enriched, and if it were like smaller, then it would be depleted. And then you also have the significance. So like the corrected p-value, let's say right here, so it should also be small for it to make sense. Okay. Okay, good. So exactly. So we also provide the hierarchical clustering implementation for you at the sample level and at the protein level. And then like a good way of visualizing it is like with a heat map. So in this case, red means low and green means high, right? Intensity. And then this means that like all of these are probably like this subset of proteins are actually like have the same behavior, so they probably like should be clustered together as they are, and these ones have the same behavior, so they should be clustered together as they are, and these ones as well. And then you also have, importantly, cluster at the protein level. So this will be at the sample level, and this will be at the protein level, and these clusters can actually then like be interpreted. Like it can be, I don't know, subcellular localization or something like this. So you have a set of hyperparameters, but don't change them if you don't have a good reason to do it. Even we will see later what they are. Exactly. So also within, I mean, within like this, the toolkit for data analysis, the principal component analysis, like procedure is very important. So what you are doing is basically rotating your data and stretching it in order to make it circular. So this is really what you're doing. So first you plot your data, let's say, then you center it as it's here, you center it, and then you actually come up with a set of axes in which your data will be circular, right? And then this is equivalent to making your covariance matrix diagonal. And then this is very important because these axes carry a lot of variance. Are you going to say?
Just maybe should we call it ellipsoidal because it's not same.
Oh yeah, yeah, exactly. I mean, I guess you can also make it circular, but not
you could. But I think this like the standard procedure in PCA is that you would leave it with different radii in the different directions.
Yeah, you're right. Actually, yeah. Sorry. Thank you. Yeah. So yeah. So sometimes actually people normalize it, but yeah, I think in proteomics is really like that exactly. So like exactly as Jürgen said, because you are actually losing a lot of information here if you change the proportion of the length. Exactly. But then this is the general idea, right? You want to find some axis in which your data, the covariance matrix of your data is diagonal, and then you do this because these are the best axes to, like, the best in the sense of they have the most variance. They represent the most variance to represent your data. Okay. Yes, exactly. And then this is what it will give you, like the proportion of variance in the number of components you take, and then the projection on each one of the axes. Exactly. You can also do, as we said before, the annotation enrichment test, and you can do it at 2 different levels, in 2 different omics. It will be like proteomics and transcriptomics, and then you have a set of, let's say, pathways that are differential, that are like differentially expressed in both, that are enriched or depleted in both, and then you have some that you can actually only see in one of them. Okay, or have the opposite behavior in one. And then there will be an example of it. Exactly. So we are slowly reaching the end of the theory, and then we will go to the hands-on. I think I have like 5 minutes more or something like this. Exactly. So this is the part of machine learning. So like, what can you use machine learning for in proteomics? Let's say one use case will be, as we said before, you have you train a model that maps expression profiles to classes. They're like green or blue here in this case, and then you expect it to generalize well to unseen data. Exactly. So the one use case will be to use this model that is already working well to get which features, so which small men will be more important. So let's say like the men are the features and the color are the classes. So like which one of like these small men are more important when predicting a class, so a color, and then this will be a candidate for markers. Exactly. So and this will be the last part of the theory. One very widely used algorithm, especially in proteomics, is the support vector machine algorithm. And the reason why this is widely used is because you can have just a few samples and many proteins, and this still works well. Normally, for machine learning algorithms, is the other way around. You need many samples, and like not more than 1% or something, like just a few features compared to the number of samples, and so you get like good accuracy. But then the support vector machines can actually handle the other case, which is more common in proteomics, where you actually have a few samples and many proteins. Okay. So exactly. So as I said, this is the end of the theory. If there are questions, we could answer them. If not, I think then we can move to the practical one. Okay.
When there is a question.
Oh, there is a question. Yes.
I wonder if we could just go over that parameter S, which gives the correction one more time.
Yes. So you mean this one? Okay. So exactly. And let me actually just, probably this one is easier. Exactly. So the variance, exactly. So this is the let's say this is without the S0. Without the S0, this is exactly what you're seeing here. So it will be like variance between groups, which mean calculate the mean in each one of the two groups, right? In this one and in this one, you calculate it. You calculate the mean, and then you calculate the difference between them, right? And then this will be your numerator. The numerator hasn't changed. And then you also have the variance, and the denominator, you have the variance within groups. which is really calculating. So you take the differences, you take the square of the difference, the differences, you sum them up, and you divide by the number of samples, okay? And then you take the square root of that. This is how like the variance between groups is calculated, okay? But then this is not the end of the story, right? Because you also have more effects that were probably not accounted for there. And then this is like, this is exactly what the zero term is about. So you have more effects, like you have more variance that is like intrinsic from your experiment and the conditions you have, and like because like the world is not perfect, right? And there is always a little bit of variability, and then you just had a random variance here, right? And this is exactly what you're doing. You are just adding a term here to account for background variability of the conditions of the sample preparation, the device operation, all of these things.